the idea of an axial core goes back to Paulson and Lemley, who published a landmark article in Cell in 1977, where they took purified human metaphase chromosomes and denatured them with polyanions, which extract histones from the chromosomes. So they dehistonize the chromosomes, and the chromosomes are thereby denatured. They deposited these on grids for transmission electron microscopy with uranyl acetate negative staining. And what they observed was quite remarkable. They saw a proteinaceous residue which exhibited the overall shape of the metaphase chromosome, the classic X shape that we all recognize from high school biology textbook. And emanating from that was a halo of loops of DNA. The idea of looping actually goes back much further in time. Johannes Ruckert was a German cytologist who in something like the 1892 was looking at the germinal vesicles of cat sharks, and he drew these stunning diagrams of sections of chromosomes from the shark germinal vesicle in which you see loops of what we now know to be chromatin. So that idea is over 100 years old. It was found to apply in metaphase in 1977 by Lemley and colleagues. And since then, there's been a lot of debate about whether or not this is an artifact of sample preparation. For instance, we see an axial core, a well-defined axial core in meiotic chromosomes, but in mitotic chromosomes, it's been less clear. It may just be local accumulation of scaffold-associated factors like the condensins and topoisomerases. But what we worked on as a graduate student with Roger at Stanford, we developed methods for studying chromosome condensation. One thing that's important to know about chromosomes, they're exquisitely sensitive to shear damage. So John Marco's group has shown this and others by stretching experiments that chromosomes will they will elastically stretch in the one nanonewton regime, but once one hits 10 or 20 nanonewtons, they undergo irreversible deformation. It's very common, nonetheless, as a sample preparation technique to mount chromosomes on slides and to apply a cover slip. But this sample preparation procedure induces flow of the meniscus, which is associated with forces in the micronewton range, even the millinewton range. And if one spent any amount of time preparing samples in this manner, one can see the chromosomes often don't survive, and one needs to look around for intact chromosomes. We wanted to avoid that, so we repurposed a parallel plate perfusion chamber onto which we can gently immobilize chromosomes with suitable surface chemistry. We can then, by means of a connected pump, freely vary the solution environment of the chromosomes, and in so doing, we can vary their condensation state, so we can condense or decondense them. And when we use this technique, actually similar work has been done by Summers in 1963, but on a more qualitative level. And when we did this, what we observed is beyond a certain level of decondensation, fibrous structure appears running down the axial length of the chromosome. And when we stain the sample with known scaffold-associated proteins, condensin 1, condensin 2, and topoisomerase 2 alpha, we see strong co-localization with that central axial fibrous structure. So that, we believe, is we think this is adds to our knowledge of the scaffold. We think it is a discrete structure rather than mere co-association of proteins along the axis. And now we're using, in the process of using electron tomography to study the sample. The problem, human chromosomes are about 1 to 2 microns thick, depending on 750 nanometers to 2 microns thick, depending on the degree of condensation, whereas electron tomography, you're limited to a thickness of around 150 nanometers. So we then need to use techniques like fib milling. We're doing this in conjunction with Maya Azubel. She's a senior scientist in Roger Kornberg's lab. And we're in the process of collecting lamellae, milling lamellae, and then subjecting them to, or collecting tilt series, which we can then use an inverse Redon transform, an approximation thereof, to reconstruct a tomogram. This is the same technique that's used to collect CT images of humans. And we're now looking at the periphery of the scaffold and trying to access central regions.
Thank you very much, Andy. That's very, very interesting. Yoav, maybe I could turn to you now, if I may, and just ask you about your approach using super resolution microscopy and why that particular method, as opposed to, say, the work that Andy's doing with cryo-electron tomography. What different kind of information does it give you, and how do you see that field advancing over time?
Right, so you can look at DNA on various size scales and different conditions. So if we define the specific problem of observing how DNA is folded and organized in 3D inside the nucleus, for example, then what you want to do is be as unperturbative as possible. So that leads you to optical microscopy, I think, would be the most likely candidate for that. You want to be specific in the sense that you would like to know what you are looking at the genomic level. You know, you don't, I mean, in this problem I just defined, you don't want to be looking at a long noodle that's folded somehow, but you don't know, what sequence you're looking at. So you want to have specificity. Ideally, you want to be compatible with live imaging at some point. Okay, so if you want to map the 3D organization in live cells, of course, that's a whole different level of complexity, but it's good to be ready for that. So I think optical microscopy makes sense. The only problem with optical microscopy is that your resolution is insufficient. If you just observe a cell in a microscope, you see a big, even if you were able to label DNA fluorescently, you would see a blur because your resolution is not good enough. But with the advent of super resolution techniques, for example, single molecule localization microscopy, which is our method of choice, you can reach resolutions of tens of nanometers routinely nowadays in the lab. So I think this combination of live cell compatibility, specificity, and now with super resolution, you can reach to the level that you can potentially map a genome, with the resolution with the genomic resolutions of tens of kb. I think that's a good approach. And another thing is, if you think about the future of this direction, mapping one genome, let's say this fantasy of just having a 3D structure of genome and exactly where each gene is, okay, in 3D. If you do it for one cell, it's a great feat, but what are you going to learn from this? So if you really want to learn something about biology, I think, and I'm saying this not as a biologist, but still, I think it makes sense, you would have to measure a lot of cells. And you have to make this approach scalable. You want to do it in a reasonable time. And so I think optics is also compatible with this idea.
Thank you so much. Shuyang, over to you. I'd be very curious to know your thoughts about where you think gene therapy and gene editing are going. As you're aware, there have been a number of safety issues recently which were quite concerning, despite the successes we've seen in diseases like hemophilia A and sickle cell disease. So I'd be curious to know where you see the future of going with gene therapy and gene replacement, and also your thoughts on somatic in utero gene editing and gene therapy, and whether you can ever see a time where we might consider treating at the germline level for diseases like sickle cell, which are so prevalent that it probably wouldn't make economic sense to continue to treat it somatically. So I've handed over to you a couple of questions there.
Thank you, Dr. Wilson. And today's talk is very inspiring. And they all look at the coding and the reconstruction in different way for I found them very, very truly inspiring for me. And for your question, I would like to start a case. The heart involvement in hereditary ATTR amyloidosis compared with to ordinary heart failure. previously, the patient presenting heart failure and arrhythmia would receive diagnosis and treatment with standard guideline. But if you ask a few more questions about the patient, like they have sensory neuropathy or autonomic dysfunction, you might realize this patient is not just a common heart failure. They could be ATTR, that's hereditary ATTR amyloidosis, a genetic disorder. In that situation, traditional therapies don't address the underlying problem because the real issue is the gene mutation lead to TTR protein accumulation throughout the body. So this is the classic diagnostic trap in rare diseases. They hide behind common symptoms, and the common treatment doesn't work effectively. For today's, the tools we are talking about AI, and really change the game. First, in today's clinical practice, AI-assisted decision tools are providing valuable early support. When I encounter a younger heart failure patient who also presents with peripheral neuropathy or a relevant family history, these tools can quickly merge multiple dimensional data and prompt me to consider an underlying genetic condition such as ATTR. Then genetic testing becomes the critical next step. It allows me to efficiently confirm whether an ATTR mutation exists, transitioning from clinical suspicion to molecular diagnosis. So once the pathogenic variant is identified, the following question emerge: can we intervene? Just like your question.
Yes.
And this is where structural biology works. Today, we cannot only find the typo in genetic code, but also see in detail how it breaks the protein structure. It's like moving beyond detecting a bug in the system to understanding why it causes the system to fail.
Thank you. And just
Okay.
very quickly, just in a very...
Yes, I think
sorry, go ahead.
about the rare diseases like sickle anemia, and we treat the patient with the gene editing, and they were treated in China.
Interesting, yeah. And just very quickly, to what extent is prenatal diagnosis being used now routinely across all of your network?
Yes. China has a big population and may have many diseases, many patients with rare diseases. And the rare diseases is not the public problem in China, also in the world. And we're screening the patients with gene and, from the hospital to hospital, and also national support with grant to give them free gene examination screening.
Yeah.
So we find many patients with the gene problem, and the patient and with families members.
Thank you. I'd like to leave some time to address the laureates, but just really quickly, just one more question to Yoav and Andrew. Just in 1 minute, Yoav. What's the prospect of us being able to visualize an entire 3D genome in one visualization in your view?
Okay, that's a great question. I had a chat about this with my colleague Gemini this morning, who is admittedly a large language model, but he came up with a very good observation. If you calculate the number of bits that are necessary to answer this question, okay, so say you have a sphere, it's 10 microns in size, and you want to know the position of, say, 1 million genomic positions with 10 nanometer cube resolution. It turns out you need about 50 megabit. And then you ask the other question, which is how many bits do I have in my CMOS camera in my lab in a single acquisition? Turns out, and I quote Gemini, it's a remarkable coincidence that it's about the same. Okay. Now, this is, of course, not the answer, because this is some information theoretical bound, right? In practice, of course, you're always limited by signal-to-noise ratio, exposure times, you know, fluorescence, intensities, and you don't want to burn the cell and so on. So the answer is, the short answer is, of course, I don't know how close we are. But I do think that computational microscopy, you know, at least it makes an interesting starting point and possibly a promising starting point. And when I say computational microscopy, let me just very briefly say what I mean. So I just, you know, told you how many bits of information there are in the system, the observation system, and in the problem that we're trying to solve. And if there's a match between these two, in principle, all you have to do is find an optical system that translates this information into something measurable. So you have to spread the photons somehow correctly, wisely, and then you have to decode this information with some algorithm. So this is a great time in history to be a computational imager, because the decoding part is done by very great algorithms that are powered by machine learning. And these are very powerful. So, you know, as the designer of the microscope, all you have to do now is figure out how to squeeze this information.
I might just move on. Thank you so much for that. I'd like to open it up a little bit. I mean, Roger, obviously we have to ask you, what excites you most at the moment in the field of 3D genome, and where do you think the field's, you know, what direction are we moving in your view?
So what I would say, knowing the work of both Yoav and Andrew very well, neither has really done justice. They're too modest in the accounts that they give of what they have done. So Yoav, in developing point spread functions and algorithms that enable the purposes that he mentioned to be achieved, really could comment further, because those have made possible rapid time-resolved analysis, such as he mentioned he would like to do. Andrew, for his part, has discovered through his own extraordinary experimental and computational skills the basis for chromosome condensation and, I believe, also decondensation for gene expression. They will both, I'm sure, talk tomorrow where they're given a greater length of time. But I think you should invite them each to spend a minute and tell you about these really transformational accomplishments.
Could you do that?
We use this system to conduct a systematic exploration of physical-chemical parameter space to understand the key determinants of chromosome condensation and decondensation in vitro. We don't necessarily know which of these determinants are the operative ones in vivo. We suspect histone acetylation has a key role and other histone post-translational modifications. But in essence, we found that our results are very well explained by a well-established theory of polyelectrolyte gel thermodynamics, which was developed by Paul Flory in the 1940s. And we adapted that model to the components of our system and performed Monte Carlo guided fitting and found a parameter set that explains essentially all of the observations that we made, including not only the well-known condensation of chromosomes with increasing ionic strength and with addition of cations of increasingly high valence. The dependence on valence is exponential. It also explains an interesting paradox, which is that chromosomes also condense at very low ionic strengths. So the ionic strength, the auto-dissociation of water sets a floor on the ionic strength in aqueous solution. One can't go below 10 to the minus 7 molar. And we find that in tens of micromolar ionic strengths, chromosomes will recondense. This was very surprising to us. But the reason we tested this was because it's a theoretical prediction that's made by the polyelectrolyte gel theory that we applied. So the group of Toyoichi Tanaka, they made foundational contributions to polyelectrolyte gel swelling thermodynamics. He was at MIT for some time. His group in 1984 published a combined theoretical experimental paper where they demonstrated this paradoxical recondensation at low ionic strength, and we tested whether it occurs in our system, and it does as well. So essentially what we found is that the chromosome is a polyelectrolyte gel, and it's a two-state gel. There's a condensed state and a decondensed state, and it can interconvert between those states through a phase transition that's unique to gels called a volume phase transition. This was predicted theoretically by Karl Duschek, a Czech physicist, in 1968, and verified by Toyoichi Tanaka a decade later. And we found that it applies as well to chromosome condensation, decondensation. Our studies have been focused on metaphase chromosomes, but we believe the same principle is probably at play in interphase, where there's also condensation and decondensation. Different regions of the interphase nucleus are in different states of condensation. This has been known since Emil Heitz's work in the 1920s and 30s on euchromatin and heterochromatin. We now have a sort of genome-wide analog of that, the A and B compartments, which emerged from the Hi-C studies of Erez Lieberman and Yoav Decker. So we think the same principle is at play there, but that work remains to be done.
Thank you. Yoav, any comments?
Yeah, sure. So for centuries, optical microscopes have been designed to serve one purpose, and that's to produce the sharpest image possible for the human observer, which made sense as long as the observer was a human. But nowadays, the direct observer of a microscope is a computer. And then the computer does some analysis, and then ultimately a human observes that result, which means that in principle, you should be designing microscopes for the computer observer, not the human observer. Okay, so what? So for example, there are certain things that normal microscopes are not very good at. And specifically, consider the example of observing a volume instead of just a thin slice. So a thin slice would be in focus, and everything else would be blurry. And if it turns out that you can modify the microscope optically by physical optical elements, that would modify the way an image appears and is recorded on the sensor in a way that encodes very efficiently a whole volume of information in a single 2D image. To do that, you need to design the point spread function. It's the impulse response of the optical system cleverly. And you also have to come up with an algorithm that would look at this, whatever strange, distorted image you measured, and would make sense out of it. Namely, would solve this inverse problem of outputting the 3D volume given the 2D image. So that's called point spread function engineering for 3D imaging. It's a lot of what we do in the lab, both the optics and the algorithms. Another variant of this problem is spectral imaging. You could think of a similar problem where you want to image multispectral data on a single optical channel. How would you do that? Well, you can also, with appropriate optical design, you can spread the information spectrally and do similar things. And let me just stress one more thing that we really make an effort to do, and that's to make our methods very spreadable and usable. We want biologists to use these tools, and since biologists have enough problems of their own, they don't need to be installing Python packages and building optical setups. So we make a point of making everything very useful. So we're now 3D printing our optical elements. We send them to our colleagues. They can do it themselves. All the algorithms, we're trying to do them one click.
I think we're actually out of time. But thank you so much to all the keynote speakers, to the panel, and of course to the audience for your listening to us and your participation. Thank you so much.